What happens when we get it wrong? Hybridization capture for microbial applications, a peek into a scary future. Hi, my name is Megan Beaudry. I'm a technical specialist in genomics at Arbor Biosciences. I'm a microbiologist by trade. Today, I'll be presenting on some of my old doctoral work that was actually done in conjunction with Arbor. I'm excited to share this snippet of my PhD work with all of you. Part 1, the problem. What happens if the microbes you thought were harmless weren't? What if a resistance gene looked rare, but only because you missed it? These are the kind of questions that keep us microbiologists and clinicians up at night. These are the reasons that we lose sleep. Let's dig into this nightmare scenario a little deeper with an example. We've collected a series of samples from hospital environments, places where our most vulnerable people spend their time. Unfortunately, healthcare-associated infections lead to approximately 75,000 deaths every year, and 1 in 3 hospitalized people will get a healthcare-associated infection. Say we are studying these environments to try to better understand why people get healthcare-associated infections and how to stop them. If we use a method that inaccurately assigns microbes, we could end up with differences like thinking something is Staphylococcus epidermis when it is actually Staphylococcus aureus. That is the difference between non-pathogenic and pathogenic, the difference between a bug and a superbug. We can take this one step further. What happens if we have inaccurate relative abundance estimates? This could mean that we are missing some of the rarest, but potentially most virulent members of our microbial community. And this isn't hypothetical. This is what happens when our sequencing tools don't tell us the full story. Today, we are going to follow the story of Mr. Mammoth, who also happens to be the Arbor Biosciences mascot. Bias can sneak into our studies by using methods that are not accurate. This means that our methods are not actually reflecting the truth. Short-read amplicon sequencing is the most commonly used method to study microbial communities today. This method is cheap, but it's heavily biased by the hypervariable region that is selected. In short, this method is similar to reading only a few chapters of a book. You don't get the whole story, which in our case could have negative implications for public health. An alternative approach is shotgun sequencing. This method is accurate, but it is expensive as you sequence everything present in the sample. This method would be like buying every single book in the bookstore when you only need a single quote. Neither method is ideal for low microbial load samples, like clinical surfaces. And that's where errors can creep in, and we can make mistakes that impact the health of ourselves and those around us. That leads us with needing a smarter way. We need something that's targeted, but can still allow for discovery. We need something that's affordable, but still powerful. We need something that's fast, but also accurate. And we need something accessible for the community, but still precise. This leads us to asking the question, what if we could focus sequencing on only what we care about? So, part 3, the solution. The hybrid capture idea. So hybridization capture works similar or like molecular fishing. It allows us to pull a needle out of a haystack, or in this case, all of the little microbes in the room with our sick Mr. Mammoth. We design short DNA probes that stick to only things we care about, and then reel them in with magnetic beads. For example, we may only care about one colored microbe, maybe the green ones. We could design probes just for those if we wanted to. The result is deeper, cleaner, and cheaper data. So how does molecular fishing actually work? How do we find that needle in the haystack? myBaits is a form of target enrichment that uses bioattenuated single-stranded RNA capture probes in a process called hybridization capture, or HybCap for short. The workflow starts by denaturing an NGS library and hybridizing overnight with the reverse complement baits, also called probes. Bait target hybrids are then bound to streptavidin-coated magnetic beads and pulled out of suspension with a magnet. Most non-target DNA is washed away, and the remaining Enrich library is amplified and taken into sequencing. We decided to take this method and apply it to microbiome studies by designing probes to the entire length of the 16S rRNA. We tested our baits against a mock community, essentially allowing us to establish ground truth, and compared them to amplicon and shotgun or unenriched sequencing, as you can see here in this lollipop chart. The y-axis represents microbial community members, and the black lines represent the true microbial community composition and abundance. The different colored dots represent the different methods. Orange is for amplicon, green is for 16S capture, and blue and purple are both shotgun sequencing. You can see that the amplicon dots are the farthest away from those black ground truth stems, whereas the enrichment is often the closest. This shows that enrichment allows for more accurate microbial community composition and abundance estimates. 16S capture allows us to capture rare members of the community, since we capture the entire length of the 16S rRNA. If we compare it to shotgun sequencing, as you can see in this table, in the red box, our on-target rate is approximately 0.1%. Now, if we look to the green box, which is where enrichment is, our on-target rate goes up to over 75%. So, if we go back to our original analogy of a book, it's like reading the entire book from start to finish. Now, what if at the same time we were looking at the microbiome, we wanted to take it one step further and look at any antimicrobial resistance genes that may be present? This is where resistome capture comes into play. With hybridization bait capture, we can do this. We can take a single NGS library that came from a single sample and enrich it for both the 16S and all antimicrobial resistance genes. We can do this at the same time, essentially not adding on any additional hands-on time, or you could do it individually from that single sample or library if you prefer. And we can do this because we've designed baits to all antimicrobial resistance genes in the Comprehensive Antibiotic Resistance Database, CARD, and Pathogenicity Island database. And this is where capture's ability to uncover rare community members really shines, in my opinion. This figure is a heat map of the known content of a custom-built mock resistance community. We have AMR cap enriched and unenriched samples on the x-axis and resistance gene classes on the y-axis. Essentially, green things do not work. Green things work very well, and red things do not. You will note in our enriched library, we have a lot of classes of resistance genes that are bright green and worked very well. In our unenriched library, we have a lot of classes of resistance genes that are red or orange and did not work nearly as well. Now let's bring this back full circle to our hospital room, where Mr. Mammoth is residing. This technique would allow us to take our research a full step further. Beyond being accurate and precise in identifying our example bacterium, we'd also be able to tell if it happens to have any resistance genes, like MRSA. This would allow us to make more informed, accurate decisions for public health. So part 3, the impact. Microbes don't stay put. They continually move from environment to environment by the way of people, surfaces, or other vectors. The intersection of human health, animal health, and environmental health are all critically important in the way that we study public health and look at our environment. The microbial communities within these spaces continue to interact with each other and can undergo things like horizontal gene transfer. Techniques like hybridization bait capture can help us trace those links accurately and efficiently. So capture helps to bridge the gap. It is more affordable than shotgun sequencing. It's less biased than amplicon sequencing, but still powerful enough to detect rare microbes and genes. As we all know, science is constantly evolving, and capture is built to evolve with those changes. First, baits have built-in pliability, allowing us to capture novel organisms that may not have been in our original design. Second, all bait sets designed for short-read sequencing also work for long-read sequencing. This helps open the door to using sequencers like PacBio or Oxford Nanopore. Third, there is no limit to the number of baits or targets that we can include in a bait set. We can design custom baits for things like bacteria, fungi, parasites, and more. In fact, we've even expanded into some of these places already by designing a bait set for 6 species of human-infecting Cryptosporidium, with the ability to capture the entire genome of each of them. We also have the ability to continually update these kits whenever it is needed by designing new kits or adding small patches to previous designs. Our end goal is simple. We want to provide comprehensive, adaptive microbial surveillance techniques that are efficient and cost-effective. Lastly, getting it right starts with having a methodology that allows us to see the problem correctly. Hybrid capture gives us a clearer, fairer picture of the microbiome and resistome. This increased accuracy can help us have a better scientific understanding and improve research and health outcomes. I want to thank you all for listening to this short webinar. Please feel free to reach out to me at mbaudry@arbordicel.com or to reach out to our sales team at sales@arbor.dicel.com if you have any questions. Thank you.